Hello and welcome to Nikolai's genetics lessons, and the first question of the day: a particular triplet of bases and the template strand of the DNA is 5' end ATG, 3' end. What would be the corresponding codon for the messenger RNA that is transcribed? And here's a variants to choose from. So imagine that this is transcription bubble. So this is double-stranded DNA. Here's a messenger RNA. This end is 5' end. Messenger RNA always grow continuously from 5' end to 3' end. So here is 3' end of the messenger RNA. And as for the DNA, double-stranded DNA, if we have 3' end here, then on the template strand of the DNA, we are going to have 5' end. And on the other corresponding strand of the DNA, we are going to have 3' end here, so 5' end here, and 3' end of the double-stranded DNA here. Now let's read the question again. If particular triplet of bases in the template strand of the DNA, so here's the template strand of the DNA, and the sequence 5' end A, G, T, and 3' end, so here's going to be our sequence A, G, and T. And here we have 5' end and 3' end of the template strand of the DNA. So again, 5' end A, G, T, 5' end A, G, T, and 3' end. So on the corresponding strand of the messenger RNA, we are going to have thymine with base pair with adenine, guanine with cytosine, and adenine with uracil. And we are going to have a stretch of 5' end A, C, U, 3' end. As you see, the correct answer is answer A. You not necessarily have to build such picture as this. We just can use another technique, so we can put right away 5' end here, and thymine would base pair in messenger RNA with adenine, guanine with cytosine, adenine with uracil, and 3' end here. And next question: in the polymerization of DNA, a phosphodiester bond is formed between a phosphate group of one nucleotide being added and which of the following atoms or molecules of the last nucleotide in the polymer? And here's the variants to choose from. I want you to take a look at this picture, and here is a sugar. This sugar has carbon number 1 here, carbon number 2 here, carbon number 3, carbon number 4, and carbon number 5. Those it looks like it is outside of this molecule, but this is a part of this molecule. So carbon number 5, as you know, DNA grows from 5' end to 3' end. So this sugar was first in this chain, and this one is the last. So 5' end and 3' end here. Each ribose has 5' end and 3' end. 5, 3, 5, and 3. So it begins with 5' end and ends with 3' end. And the other strand of the double-stranded DNA is just going to be antiparallel. It also goes from 5' end direction to 3' end direction. So we call these strands of the double-stranded DNA being antiparallel. One more time. In the polymerization of the DNA, a phosphodiester bond is formed between a phosphate group of the nucleotide being added and which of the following atoms or molecules of the last nucleotide in the polymer. And as you remember, each sugar, which is ribose, so here is a 3' end and here is a 5' end. This is beginning, this is the last one, and to this last one, and you phosphodiester bond would form, and attached to the carbon number 3, we are going to find a group which is OH. So would be attached to the carbon number 3. And as you see, this is answer A. And this is all for today. Subscribe and see you in the next video. Goodbye.